So AGTC is a clinical stage gene therapy company focused on orphan diseases of the retina, specifically monogenic diseases that can be treated with AAV. The company has a clear strategy and tactical plan to become a world leader in this area, and this is predicated on being established by a group of founders from the University of Florida with significant expertise in this area, and subsequently 15 years of work by the company to develop a leading gene therapy platform, which includes optimization of the AAV vectors for safe and sustained delivery, as well as developing a cutting-edge manufacturing platform. The company has a track record of progress, including to date treating over 100 patients in clinical trials. It has extensive expertise within the management team, understanding all aspects of vector selection, design, manufacturing, and delivery. The company has a significant IP portfolio with over 100 patents protecting all aspects of the gene therapy requirements, capsid, regulatory amendments, manufacturing, and so on. As I'll describe in the next few slides, we have a broad pipeline of programs with the potential to deliver significant value to patients over the next 2 to 3 years, together with a product engine based in research and development labs in Alachua, Florida, and also recently in Cambridge, Massachusetts. Importantly, earlier this year, we signed a research and development partnership with Biogen, which matches AGTC's AAV expertise with Biogen's biologics development and commercialization expertise. So this is a snapshot of our current pipeline. What I'll be doing today is describing just the top 4 of those, which are either in clinical development or nearing clinical development. But as you can see, we have a number of other programs in preclinical development, and we continue to add new programs to the portfolio. So moving on to the lead program, X-linked retinoschisis. This is caused by mutations in a protein retinoschisin, which results in the schisis or cyst formation and splitting of the retina, ultimately leading to poor vision, which is not correctable by eyeglasses. There are multiple mouse knockout models which have been used to show proof of concept, meaning that the human protein can be introduced and to correct the deficits either in the outer nucleus or the photoreceptors, or to also show the expression of the protein in itself. An IND for this program was filed in 2015, and we're currently in a phase 1/2 study, which is shown here. So this is an ascending dose study with the aim of defining a maximum tolerated dose, at which point both adult and pediatric patients will be enrolled for a cohort expansion. The endpoints are primarily safety, with secondary endpoints of visual acuity, visual fields, ERG, and OCT. A total of 27 patients will be enrolled in this study, and the progress has been acknowledged with the first milestone earned from the Biogen collaboration. The second 2 programs are in achromatopsia, B3 and A3. So achromatopsia is a deficit in cone receptor function, resulting in severe impaired vision and day blindness. Mutations in the cyclic nucleotide-gated protein subunits B3 and A3 account for about 75% of the cases of this disease. Fortunately, there are both dog and sheep models that have the same genetic mutations and defects in humans, and we've utilized those in proof of concept studies. So on the top right here, we're looking at a maze transition time experiment where dogs, which are normal, essentially take limited time at different light intensities to travel through the maze. For the untreated animals, you can see they take significantly longer, and in fact, as would be expected, the duration is even greater in brighter light. Following AAV therapy, however, we were able to return the transit times to almost normal at all light levels. We have taken this program now into non-human primates, and we've developed a vector which effectively delivers the protein, either B3 or A3, to the cones, both red, green, and blue cones of the animals. Recently, for the B3 program, we filed an IND. The fourth program is X-linked retinitis pigmentosa. So approximately 15% of RP is caused by mutations in the RPGR protein, and about 70% of those are linked to the X-linked form of the disease. Mutations in this protein cause a loss of function of the protein, and as a result, blindness and progressive constriction of visual fields, with approximately half of the patients being legally blind by the age of 45. There is a genetic dog model of this disease, and we have shown by reintroducing the human protein or the canine protein into the dog that we can rescue function, and by this I mean the reduction in the photoreceptor layer as well as the recovery of both rod and cone EEG response. We have also now developed a primate vector, or a vector which is effective at delivering subretinally this protein RPGR to both rods and cones, and the figure shows here an improvement over the original vector that was published from an academic lab. So one of the advantages we believe the company has is in its manufacturing technology. So we have improved the process to increase the yield approximately 25 to 30-fold, improving its cost effectiveness as well as the time to produce the material for both preclinical and clinical testing. In addition, we're able to fully characterize the AAV product candidates in this assay, and also show that we're able to clear entirely the herpes simplex virus, which is used to produce the AAV. We've taken this to a commercial scale and also transferred it to both partners and contract manufacturing organizations. The platform itself is fairly flexible. We have multiple serotypes and transgenes that we've prepared using this platform, and currently have both recombinant AAV1 and 2 in the clinic. And lastly, the platform has been approved by both the FDA and the EMA for use in clinical trials. So to sum up, this is a 2-year time frame of the key important milestones that are coming up for the company. In 2016, we intend filing an IND for the Achromatopsia A3 program. We will be getting the initial clinical data from both the XLRS and the B3 program in 2016. And then in 2017, we'll have 1-year data for both the XLRS and the B3 program. We should also have the initial clinical data for the A3 program, and we intend to file the IND for the XLRP program. Thank you for your attention.